Hello, this is a discussion of Chapter 15, Microbial Mechanisms of Pathogenicity. All right, so this little chunk, we're going to talk about the principal portals of entry for pathogenic microorganisms, ID50, which is infectious dose, and LD50, which is lethal dose. All right, we're going to explain how microbes adhere onto the host cells. So some definitions: pathogenicity, the ability to generate sickness. All right, and then virulence is the degree of pathogenicity. All right, we've got portals of entry. The most popular is the mucous membranes. Skin is usually really nice barrier unless you have a hole poked into you, and we call that the parenteral route. So if you nurses are giving injections, be careful. Bites, wounds, cuts, surgery. So most pathogens have a preferred portal of entry, right? Respiratory diseases are going to go into the nose or the mouth, for example. All right, so mucous membranes, pretty much all your openings are covered with mucous membranes. Respiratory tract, the digestive canal, the genital system, the urinary system, and conjunctiva. Remember, that is the lining of your eyes, basically your eyelids, and then it goes a little bit over your eye. All right, so the most commonly used portal of entry is the respiratory tract by inhalation, and everyone has experience with that. COVID, for example, was able to access your body through the respiratory tract. All right, then we have the digestive canal. So anything you eat, drink, or if you put your fingers in your mouth, you can pick up germs. Then we have the genitals. So any STIs are going to be accessed by the genitals. But keep in mind, sorry to bring this up, but HPV, for example, can actually be spread by other means than just direct genital to genital. So keep that in mind. All right, so your skin, luckily, when your skin is not broken, it is impenetrable by most organisms. But here is the exception. We do have openings. So hair follicles and sweat glands and ducts can easily be infected by microorganisms. All right, so parenteral route. Microorganisms are deposited directly into the tissues when you get a hole poked in there. A cut, a puncture, ooh, I hope you don't get a gunshot, a bite, surgery, or injections. Those are all called parenteral. I think parenteral pokes through your skin. All right, some numbers. How do we know how bad a microbe is? Well, we have two measures. So ID50 means the infectious dose, where 50% of a sample population will get sick. Okay, so for different microbes, you might need more or less. Example, Shigella, maybe just 10 little bacteria could get you sick, whereas you might need hundreds for a different type of bacteria. Then we have the lethal dose 50, which is the lethal dose where 50% of a sample population will die. Measures the potency of a toxin. All right, so here's an example. Good old Bacillus anthracis. The different routes of infection require different numbers. So ID50 for the skin, if it pokes into your skin, it is a lot less than if you are going to eat it. Okay, you're going to need a lot more microbes, individual bacteria, in order to get sick. All right, then we have the LD50. Now, the lower this number is, the more deadly the toxin is. So out of these three, and you want to look at the units really carefully. So this is nanograms per kilograms. Remember, nano is times 10 to the minus 9. So that's a really small amount. Okay, ch15, you need more to kill somebody. Staphylococcal enterotoxin, entero means intestines, you need even more. So the most deadly one here is botulinum. So adherence is how it's going to stick on. Just think of an adhesive bandage. We have different little things that are going to stick on. We have adhesins, ligands. A glycocalyx is a sugary goo. So one really good example is Streptococcus mutans. So they have dextrin that is going to stick the bacteria to your teeth. Why, unfortunately, we have to go to the dentist. All right, then we have fimbriae, which are little hair-like things. So they're going to stick on. So an example, actinomyces is going to adhere to the glycocalyx of the S mutans. This is why we have to get our teeth cleaned, right? Once you have one type of bacteria, other ones are going to stick to it. And I'm sure many people heard about the viral spikes during the major pandemic we all went through. So remember the spike, remember viruses have an envelope, some have an envelope and some don't, and then they have a spike on the outside. All right, so this is the virus, and then you will have a host cell, will have a receptor, and the spike will just go right into the receptor, and that's how it adheres onto your human cell, for example. All right, so again, adherence, you can see this is the little spike protein. It's in our phospholipid bilayer. The purple thing is made out of protein. And then we have E. coli, and this is probably too little for me to see, but I think it's staph. All right, so 3 portals of entry. How do things get into your body, right? Pretty much any opening will allow microbes to gain access. So respiratory, genital, parenteral are 3 examples. All right, let's look at this one. The LD50 of botulinum toxin is 0.03 nanograms per kilogram. The LD50 of salmonella toxin is 12 milligrams per kilogram. Which is the more potent toxin? Okay, what do you think? That's right. No, I hope you got it right. This is going to be the more potent because remember, it's nanograms. So you only need a tiny bit of botulinum toxin to kill someone. Please don't do that, but just saying. All right, how would a drug that binds mannose on human cells affect a pathogenic bacteria? Well, remember that some bacteria do bind onto the mannose. So if the drug covers up the mannose, then it would not be able to adhere onto your cells and you hopefully would not get sick. All right, so this little section, we're going to talk about capsules and cell wall components contributing to pathogenicity. We're going to look at some enzymes, okay? Coagulase, kinase, hyaluronidase, collagen ACE. Look at antigenic variation, how bacteria such as Listeria monocytogenes use the cytoskeleton to enter the cell, and then how microbes avoid destruction by phagocytosis. All right, so capsules we've already learned about a little bit, right? Remember we have the cell membrane and then we have the cell wall. And then around the cell wall, we have the capsule on the outside. So if your microbe can hide from the immune system with the capsule, it won't be able to be seen and it won't be eaten up. So examples of microbes that have capsules: Streptococcus pneumoniae, Haemophilus influenzae. Now, this is not the flu, right? Remember, the flu is a virus. So this is a bacteria, and we do have a vaccine for it. Bacillus anthracis and Yersinia pestis all have capsules that keep them from being devoured by cell-eating phagocytosis. All right, now we have cell wall components. The M protein is going to resist phagocytosis. So an example of a pathogen that has M protein is Streptococcus pyogenes. Then we have good old gonorrhea, Neisseria gonorrhoeae. So the OPA protein is going to allow that to attach to the cells here. You see it? And then we have a waxy lipid. I know you have an acid-fast stain lab online. So the reason we have to do the acid-fast stain is because it's harder to penetrate these cell walls due to the mycolic acid, which is a waxy lipid. So example is Mycobacterium tuberculosis. They get into the phagocytes, but then they are not digested. All right, so let's look at these enzymes. All right, enzymes, sometimes they're good for us, but unfortunately these bacterial enzymes are pretty scary. So we have coagulases. They are going to clot or coagulate the fibrinogen, and that's going to form fibrin. So that's what your body normally does when you're bleeding. It's okay, but you don't want blood clots forming if you're not bleeding. All right, then we have kinases. Of course, we see ASE. It's a good guess. It's an enzyme. It's going to digest fibrin clots. All right, then we have hyaluronidase. I'm sure you've heard of hyaluronic acid. If you look at any lotions and whatnot that are supposed to prevent wrinkles, sometimes they will have hyaluronidase in them. It's kind of a fake out because it's not going to be absorbed right into your skin, but it's a good effort. Okay, so hyaluronic acid is a host polysaccharide that holds cells together. All right, and then collagen is also part of your body, right? Part of your skin. So these enzymes, it's pretty scary to me. They have collagenase that can actually break down the collagen in your skin and in your tissues and just get right into your body. Now, gonorrhea, we've had it for a long time, right? It's been with humans for history. How in the world is it just keep on surviving and we don't develop immunity? Well, the trick is that it has IgA proteases. So remember, IgA is immunoglobulin A, and that is just another word for your antibodies. So IgA would be in your mucous membranes, so it would be in the genitals where gonorrhea gets in there, but gonorrhea is super sneaky and it just cuts up those antibodies and gets right into your body. Okay, another way that microbes evade your immune system is antigenic variation. So your body might have created some Igs, immunoglobulin, whether that's A, G, M, D, etc. GAMED spells gamed, right? IgG, IgA, IgM, IgE, IgD. All right, so your body may have created an antibody which would have blocked this pathogen. However, instead of having these red pili, now it has these green pili. So now this antibody can no longer recognize that antigen and it escapes detection by your immune system. All right, and then how do these guys actually get into your body? the surface proteins are going to be produced, and they are going to actually rearrange the actin filaments. So remember that actin filaments are going to be just kind of right under the cell membrane. You're going to have, I'm going to use purple for the actin filaments. They're going to be under the cell membrane. Remember we looked at the video with the cytoskeleton? So actin filaments are part of the cytoskeleton. So it's going to rearrange and cause membrane ruffling. It's going to change the surface of the cell membrane, and then they're just going to be able to hop right in there. Okay, they use actin to move from one cell to the next. All right, they can also survive inside the phagocytes. So there's a low pH. They can survive in the low pH. Then another thing they can do is escape from the phagosome before it actually fuses with the lysosome. Remember, just think of Lysol. The lysosomes are going to clean up. They have acid and enzymes to kill the bacteria. So next thing they can do is prevent the fusion of the lysosome with the phagosome. Okay, so this shows you those ruffling, just like the potato chip, right? Ruffles. And then the little flagellates holding on, and then you can see these guys are just getting right in there. All right, I posted this with the video so you can watch them as well. All right, biofilms, really important as nurses that you don't create biofilms on people, right? So biofilms are going to make it really hard to attack the pathogen because they're going to be coated in an extracellular polymeric substance, which is basically some goo, sugary goo, and it's going to protect those microbes from antibiotics and disinfectants. Okay, so what do capsules and M proteins have in common? They are going to resist phagocytosis. Okay, would you expect a bacterium to make coagulase and kinase simultaneously? No, because coagulase is going to clot your blood and kinase is going to dissolve blood clots. So you would expect one or the other. All right, many vaccines provide years of influenza of protection. Why doesn't the influenza vaccine offer more than a few months of protection? Because that guy is super sneaky. Remember, it's going to do that antigenic variation. It's going to change the surface molecules. It's antigenic variation. So without those surface molecules being the same, remember your antibodies are not going to be able to bind them. Okay, how does E. coli cause membrane ruffling? It's those actin filaments.